I want to move on now to our last session for today, which is the interface between microbes and neuroscience. And our first speaker today is Nancy Moran.
Hi there. Can you hear me? Great, thanks. So we're going to have another visit to an unusual system. And I'm not going to talk really much about neuroscience, but I'll give a little bit about behavior, because honeybees, in fact, are a system where there's been a lot of behavioral work in neuroscience. And so the potential with this system, I think, is really great for behavior. But first, I'm going to be introducing you to the system. It's a system that I started really working on about 6 years ago with the aim of turning it into a system, a microbiota, that could be studied as an experimental system where we could look at many different aspects of how a microbiome is assembled, how microbes interact within it, and it has some advantages that I'll be explaining to you. So here's a rough outline. First, I'll introduce you to the system, talk about some of the experimental approaches we can use, the behavioral model that bees comprise, and then give a little example of some experiments. We're just really starting this kind of work of the effects of microbiota on bee behavior. And then at the end, some issues with using bee microbiota as a model. So just to introduce you to honeybees, pretty much everyone reads about honeybees in the news all the time, so probably you all know this, but they're a social insect. They form very large colonies. They're dominated by female workers. There can be from 30,000 to 100,000 workers in a colony. They're the most important agricultural pollinators. So they're present worldwide. They're basically a domesticated species that sometimes escapes, but they're present all over the world and have been bred for use for honey production and pollination. And in the last approximately 10 years, there's been a lot of attention to problems with colony health that you've probably heard about. Colonies dying, the so-called colony collapse disorder, all kinds of pathogens and problems, insecticides and so on that are affecting them. And it's a big economic problem. And they have a distinctive gut microbiota that I'll talk about. This just shows let's see, go back one. Last year, there was a White House report on the importance of pollinators and all the problems with them, and an invitation to the various government agencies to come together and try to take measures that will help pollinator health, including primarily honeybees, but also wild pollinators. Okay, so the gut microbiota, this is just a kind of summary of the organization of the gut microbiota. It's primarily in the hindgut, so the distal gut contains 99% of the microbes in the gut, and that's comprised of two different sections, the so-called ileum, which is not really equivalent to the ileum in mammals, but the ileum and the rectum. Okay, so most absorption occurs in the midgut, but the bacteria are mostly back here, and some absorption occurs down here. There's approximately 9 species that make up 99% or at least 95% of what's in your normal honeybee. So any honeybee anywhere in the world that you collect will have these 9 species in it, and they do not occur outside of honeybees, with one exception. One of them, this Acetobacter can be found also outside in nectar. They're gut organisms. And so in many ways, that is quite similar to what we see in the mammalian gut. The things that live in our gut, for the most part, only live in the gut. We don't find them in our food. We don't find them in the environment. They live in our guts. And the same is true for honeybees. These are specialized for living in the guts of bees. And they form these very dense and organized communities within the gut, approximately 10 to the 8th or 10 to the 9th bacteria per bee, which is actually quite a dense community. This is the development of the community over the lifespan of a bee. So here's the egg is laid by the queen. There's a pupal stage that's in the hive in these little hexagonal cells. They're fed by other worker bees. There's actually very few bacteria in these, and they tend to look more like environmental bacteria and to be very erratic. The pupae, the larval stages of honeybees have relatively small communities. Then there's the pupal stage, and this is really key to our experimental approach. At the pupal stage, the pupa forms, basically the entire gut is turned inside out, and everything it leaves the body. So the pupa, basically when the adult emerges from the pupal stage, it's germ-free. And so this gives us a ready supply of germ-free organisms just from your normal hive. And so we use the trick of taking the pupae out when they're still in the capped pupal cell, allowing them to emerge in the lab where we have germ-free individuals that we can then manipulate as an experimental system. Okay, so for the most part, you get this long-lasting community that's about 10 to the 9th cells. There is variation among individuals. There's 8 or 9 species that make up almost everything in here. There's a lot of strain variation within those species, so it's not quite as simple as I thought when I first got into this system. When you start really looking at the genomes, each of those species can have many different strains. A single honeybee, for example, one of the species we've looked at a lot called Guillemella apicola, can have up to 100 strains even within one bee. So it's more complicated than the small number of species would suggest, but it's dominated by these species. Sometimes this microbiota is disrupted, so we get some individuals, stressed individuals, ones that have pathogens of different kinds. So there are viral pathogens that are very important in bees, as well as some trypanosomatids that are important. Some of those individuals, or very old individuals, will have a disrupted microbiome, and in those, they often get a lot of Enterobacteriaceae, including Serratia and Klebsiella, and actually some of the same thing groups that can be pathogenic or a sign of dysbiosis in the human microbiota. So what are the similarities and differences? The gut microbiota in bees and humans are dominated by specialized co-evolved lineages that live in their host. They're specialized to live there. They're not just environmental organisms that have been collected by the host in a selective way. They're actually only living there. They live under low oxygen conditions. So one reason that no one had studied the main reason no one had studied the microbiota of honeybees, because honeybees are studied a lot over the decades, is that no one had tried growing them under the right conditions. So only when we had sequence-based syntheses, we realized the things that people had grown on plates were less than 1% of what was really there. The main things there grow under mostly microaerophilics. A couple of them are actually anaerobic conditions, and some of them are facultative anaerobes, but they don't grow well under full oxygen. They're socially transmitted, so just like in mammals, bees get them from their relatives, their family groups for the most part, and maybe from other individuals they encounter later in life. So they either get them from the surfaces of their house, the hive, or directly from the hind end of another bee. And the beekeepers didn't like this because bees are supposed to be clean. They don't defecate in their colony. But then I just point out that's actually how humans get our microbiota as well, and it's not considered that bad a thing. So basically, it's a fecal route for the most part. There's a high level of strain diversity. Oops, I keep doing the wrong button here. There's a high level of strain diversity within the species, just like in the human gut, where we get really a lot of microdiversity of lineages. There's a mixture of gram-negative and gram-positive bacteria. There are no archaea in the bee gut, at least not normally. They occur mostly in the distal gut. So just like in humans, they're in the distal gut after most nutrients have been absorbed. So there's a lot of complex plant polymers there and that are still available to be digested, but the easy stuff has already been absorbed. So it's actually a nutrient-poor environment. You get some individuals with dysbiosis, like old hosts, or following antibiotic treatment also. And then you get these things like Serratia, Klebsiella, and so on being more common. The host immune system does modulate community composition. I'm not going to talk about that, but of course they only have the innate immune system, but it is important. There are antimicrobial peptides produced in epithelial cells, and the bacterium living right on the surface is most resistant to those, and it seems to mediate the ability of others to colonize. There's a history of antibiotic treatment, just like in humans, so there's a lot of antibiotic resistance due to that history, because in apiculture, beekeepers use antibiotics, at least in the US. Differences, well, there's only a much smaller number of species present than in the human gut, of course. And another main difference, and one reason that this is an attractive system that I'm continuing to develop, is that we can grow all of the species in pure culture in the lab. So we have, basically played around with it, and I had a brilliant grad student, Walden Kwong, who's now at University of British Columbia as a postdoc, who basically mastered it. We can grow all of them in the lab. And obviously, it's cut off at the bottom, but obviously there are differences as well, right? There's no adaptive immune system. There's differences obviously there are many differences between bees and mammals. So here's our general strategy for studying these. We have our newly emerged adult bees, and mostly we work with the honeybee. I'll just mention that bumblebees, which are related to honeybees, they diverged 50 million years ago or so, they also have the same sort of species of bacteria, the same lineages that are bee-specific. So these bacteria have evolved with social bees for millions of years and are present in different species of social bees. Of course, there are different strains in different species. We mostly work with honeybees, but bumblebees have some advantages as well. We let them emerge in the lab. They're germ-free, and we've validated this many times. They're basically completely clean. And then we inoculate them with known strains or combinations of strains. We have some ability to engineer these so we can put fluorescence genes or luminescence genes in them, monitor the composition, the interaction within hosts, and then we can monitor the effects on the host phenotypes, including behaviors. And we inoculate them primarily just by feeding them. We can feed them either in groups with a trough, or we can hand feed them to give them a known amount of in a sucrose solution. So in sucrose solution, or it should say, or on pollen. This is just some of the images from what we do. So we actually grow them in outdoor hives. So we basically collect the frames from the hive that we have on the top of a building at UT Austin. We've got about 10 colonies up there. We pull the pupae from the frames. We have to do it at the right stage before they've started chewing out, because they otherwise will acquire some bacteria. This shows the different stages of the pupae, so we have to get them before they're really getting dark like that. Keep them in a dish in the lab. The temperature inside a beehive is 35 centigrade. That's really close to the 37 that we keep incubators at normally. So keep them at this temperature, they emerge, and then we have our germ-free bees that we can use in different kinds of experiments. There are also protocols, we don't use them yet, but there are protocols for in vitro rearing of bee larvae that are used by quite a few labs around the world. And those would also be a way to perhaps reduce variation. They'd also be a way to make them more abnormal, which is one reason I don't use them. At least the ones we use had a more or less normal sort of larval existence. And so we're starting with those but this could be useful for some kinds of experiments as well. And this is just to show bumblebees because they're really adorable things, but we also use bumblebees. You can basically have a bowl. The colony is just a thing that you can carry around. It's very small, maybe 100 individuals in it. And they're useful also for some kinds of experiments. And they're commercially available because they're used in the pollination industry, and all those blueberries that you can buy nowadays. Bumblebees pollinated every one of those blueberries. And this shows the results of colonization, a natural versus experimental colonization. So here we have a natural one in in the hive. We can see this is Snodgrassella alvi. So all of these bacteria had no names, were nothing before we've basically grown all of them and named all of them as a big part of what we've done the last few years. So this just shows, this is a species that forms a very solid layer on the wall in the ileum. So the ileum has these longitudinal folds in it, and you can see the purple here is a Snodgrassella, and then over here is just a lab inoculation, just showing that you get this very robust inoculation. A major focus that we have is, why does Snodgrassella get to colonize the gut? Even a very tiny number of cells, Walden Kwong estimated that fewer than 50 cells of Snodgrassella can result in a full colonization. So there's something the host recognizes, they both say yes, and then it just takes off and forms a very large population, whereas most bacteria cannot colonize there. And this shows a dual infection in the lab with Snodgrassella alvi, Guillemella, and then, so Guillemella form these little clusters of cells. They're the red ones. Snodgrassella is this more continuous one here. So, and it seems that Snodgrassella has to colonize in order for Guillemella to be able to. We've done a lot looking at their metabolic interactions using genomics and metabolomics. Basically, they seem to cross-feed a lot, and they're interdependent on each other. Those aspects I'm not going to talk about more. But one major interest in this system is just to be able to look at the interactions of organisms that have co-evolved. A lot of those interactions also are antagonistic. They're basically, there's a war going on in there, and there's all kinds of toxins, type 6 secretion systems for killing other bacteria, and that's a big part of kind of the dynamics in this community and in the human community as well, I would say. So we can also we can just compare germ-free bees with conventionalized bees, and so we can basically reconstruct what looks like indistinguishable from a normal natural community by just inoculating with homogenized hindguts of bees from a hive, and then we can get a conventionalized microbiota, compare effects to how the phenotype differs from germ-free bees. And there are a lot of effects, the morphology of the gut, the the metabolism, a lot of aspects of bee biology is affected by the presence of the gut microbiota. And so I've been working with a cluster of people. We got some DARPA funding for, it's called the BRICS program. So basically it's to manipulate a community to genetically engineer members of a naturally occurring community to affect something else. And we use the bee microbiota as kind of a system to do this in. And so I'm working with Andy Ellington and Jeff Barrick and Brian Davies and Hal Alper at University of Texas, Austin. They're all kind of doing tricks with the bee microbiota, which has been great, and we've made a ton of progress on being able to do this. And the idea is that we can sort of design communities, put them in the bee, and then see what happens over time. And this general experimental approach has enabled a lot of kinds of studies. I just put a few examples up here to give an idea of what we've done. We've been looking at genomics and genetics, metabolism, antibiotic resistance. For example, one of the bee gut microbes, Gilliamella apicola, which is it's a gamma proteobacterium, has a huge ability to metabolize sugars, different plant products, and sugars and polysaccharides, and that potentially is very important in bee biology and enabling them to deal with different compounds. A lot of these sugars are actually toxic to bees directly. So this is just to give an idea of the range of things that have been done. This showing, so for example, some strains of Gilliamella can break down pectin, this complex plant polysaccharide. When it's broken down, it releases a bunch of sugars, which are sort of non-standard sugars that animals generally can't digest. But these bacteria have the ability to use these sugars. And there's a huge amount of strain variation. This is a very dynamic set of genes, so different strains can use different sugars, break down different plant polysaccharides, and so on. So this is actually very similar to what goes on in the human hindgut or colon, where plant polysaccharides can be broken down by different strains of bacteria. They differ a lot in which ones they can use, and potentially this can be important in nutrition for the host. Okay, so as I mentioned, we've been working a lot on engineering the bee gut microbiota. We can grow them all. They don't grow super fast. They're not as easy to work with as E. coli or something, but they're not terrible. We can grow them on plates. And we've managed to develop a couple of different vectors for introducing genes into them so that we can add pathways to these bacteria and see what they do. So we can add fluorescent markers or other metabolic pathways. We also have, from Brian Davies' lab, ability to put in protein secretion apparatus into the bacterial, into the gram-negative members at least, and so that they can secrete different. Then we can add a protein that has the right signal on it to be secreted. So basically, we can secrete peptides or small molecules, and we can sort of mark them just for visualizing changes in the communities. So we've been doing a lot of work with that, and this is just to show, here's in the ileum of a bee, you can actually see individual bacterial cells. These big things are the pollen grains, which are autofluorescent, and here's a luminescent marker put into, there's a native serraceous strain from bees, and you can actually visualize it through, even in a live bee, at some level. So we got interested as part of this DARPA-funded project in introducing compounds that would have an effect on the behavior of bees. And so one of the first ones we tried, which I'll tell you about as an example, is dopamine, or L-DOPA, the precursor to dopamine. We introduced that pathway onto one of these plasmids, put it into one of the bee gut bacteria, and then looked at the effects on the bee behavior. Bees as a behavioral model are actually pretty well developed. A lot of people have studied bee behavior. You probably all know about the waggle dance. Well, there's a whole lot of behavioral work on bees, and this has been led by Gene Robinson at University of Illinois, and numerous others have done really pioneering work. They have complex behaviors because they are social. They interact with each other. They have complex tasks during their lifetime. So they have sort of more complexity in their behavior than many insects have. So there's a lot of literature on it. And I'll point out that many of the small molecules that control bee behavior are the same between bees and mammals. They're conserved across animals. So neurotransmitters and many hormones are largely conserved, and also in worms. There's a lot of behavioral effects of these compounds that are documented in bees. So some of these are very simple behaviors. Some of them are more complex. For example, learning. to associate a cue with a condition, gustatory responses, activity level, buzzing response, anger. So this just shows a couple of behaviors. So one is this proboscis extension response. You can see how readily the bee responds to a concentration of a compound or sugar or else some other some odorant, and see whether it does this. So there's a lot of different behaviors that are easy to score. So looking at dopamine, some previous results had already shown first that these are from other previous studies that feeding L-DOPA to bees increases the amount of dopamine in the hemolymph. There is no blood-brain barrier in bees, so that should increase the amount getting to the brain. A dopamine antagonist diminishes the ability to associate a scent with a punishment, and then bees fed dopamine or L-DOPA can learn to associate a color with a punishment faster than control. So some of these wires cause a shock, some don't. They learn which is better to sit on, yellow or blue, and then you can monitor that. So we've done actually this yellow-blue type study, but I'll show you another kind of study. This is another aversive training technique, and I'll just show you this as an example. So basically, you present them with a scent. You observe whether the sting, 3 seconds, whether the sting is extended, and then whether it is or not, they get shocked at second 4, and then they rest, and then you do that again. And so you do this multiple trials, and you see whether they learn to evert the sting before the actual shock occurs. Let's see. So now I don't know if maybe you need to, yeah, thank you. So this is just a little movie to show the training thing. Oh, here's an untrained subject. So here's the scent, no sting, no sting. Now the shock is applied, and there's this, you can see the stinger if you're looking really closely. And here's one, a trained subject. Here's the scent. You see the stinger is already out even before the shock is applied. Then it goes on and the shock is applied. So that's basically the protocol. And we tried this after comparing bees that had an L-DOPA producing gut symbiont to bees that had the same symbiont but producing just GFP or something as a control, and wanted to see whether or not we could get an effect of the symbiont. So basically all the steps are there. It should actually work, and in fact it actually does seem to work. So this is the beginning of our results. So basically the blue here are the ones that have the ones producing L-DOPA, and they are learning faster than these here, which are the controls, which are learning but not very quickly. So they seem to learn faster. And then if tested 1 hour later, so you leave them alone for 1 hour, and they also remember better. So they remember and have a response more often than the controls. So it seems as though a symbiont producing this neuroactive amine can have an effect on behavior, just as a proof of principle. And actually, it's not terribly surprising all of the steps were there, but it does mean that it's actually taken up in the gut and in sufficient amounts that it can actually affect behavior. So we have ongoing studies of different things. So the L-DOPA, we're still doing some work with that, but we're looking at various other compounds as well, and also just different hormones. So other than, for example, insulin, we have this protein secretion system that we can put in the symbionts and then secrete different proteins. So this is Brian Davies' sort of corner of the project, and he has this working well. And so we want to be able to look at how that affects insulin as an active hormone in bees as well as mammals. And then this is an alarm pheromone that is produced in bees and causes them to be angry. Okay, so just at the end, I want to include the 2 slides, one on animal welfare in the context of research on bees. So as an insect, it's not covered under any of the policies or any laws. So just like worms, there's really no rules about it. We don't have to worry about that. We try to avoid things that might make the bees suffer. We try to kill them quickly. For example, at the end of experiments, we freeze them and they die within 2 minutes. That's a very common way for bees to die in nature. And so, and there's really a lot of work on bees as a domestic, agriculturally important species. So there's many, many labs that work on bees in the context of their role in agriculture and so on, and also as behavioral subjects. So there's a lot of protocols for using bees. And then a few challenges. Well, honeybees, they're complicated organisms. In some ways that makes them interesting, but it also makes them hard to work on. They have these complex social lives within large colonies. When we are studying them, we're moving them from the colony and mostly studying them in the lab. We do some studies where we put them back, not with the GMOs, but where we put them back in the colonies and look at their survivorship and so on. But mostly we study them in the lab, which is highly artificial. We don't see the full normal phenotype of a bee in the lab because their queen isn't there. All kinds of things are not normal. So that's a challenge. We haven't been able to establish germ-free colonies. We tried it, but it didn't work. Basically, they acquire their microbiota as soon as they emerge in the colony. We tried getting a germ-free queen and some germ-free workers, putting them together and trying to tell them to be a colony, but there's a lot that goes on to becoming an educated bee in the colony, and we weren't, they didn't know how to take care of their queen. They didn't know how to do things. They were just a complete mess, the ones that had their early life in the lab. So I think that's a challenge to actually get we can give antibiotics to a colony and reduce the microbiota, but we can't clear it by any means. There's a lot of variation, so there is a lot of genetic variation in honeybees. There's all these different breeds that have been selected, through artificial selection around the world over time. And there's also a lot of age-associated variation, which we can control for at some level, but because there's a lot of knowledge about how the behavior of bees changes with their age. They have different tasks and different behaviors with age. So those are sources of variation that you have to be concerned about. And there are a lot of homologies in hormonal systems, immune systems, and nervous systems across animals in general, as Angela pointed out earlier. But obviously, many aspects of human biology do not apply to bees. I mean, there's just a lot of differences as well. So there's many things about the human microbiome that you can't study by studying bees. One motivation for studying bees is actually the bees themselves. They're important, and a lot of them are dying. So we are hopeful that some of what we find out will actually be helpful in improving the health of bees as pollinators. And finally, I've just listed here really the people who directly did work on some of the things I talked about. So Andy Ellington, he's the crazy man who came up with the idea of applying for this DARPA money and using the bee system to do this, which has really turned out great. It's been a lot of fun, and we've made really fast progress. Jeff Barrick, who did a lot of the genetic engineering and basically holds everything together. Sean Leonard, a wonderful graduate student at UT Austin, who did a whole bunch of building the plasmids. He's basically the main person behind that. Eli Powell, a technician who basically, I told him we have to study behavior, and so he went in and read the papers. We got all these assays going, and it was really out of the blue for us, and he's been really great at getting that done. Walden's the first person to get the bacteria in culture, and all the others contributed in some way to different aspects that I talked about. So thank you.